Hey, thanks for joining me today. I'm going to be tying together a few loose ends about how nerve cells communicate with each other. That means we're going to be learning about saltatory conduction and synapses. Now, nothing gets your synapses firing more than learning about themselves, so buckle up because it's going to be one wild ride. Now, before I get back into the details, I want to remind you of a general overview about how the nervous system sends signals. It's really alternating between electric and chemical signals. Electric signals would be the action potentials that travel within a single neuron, and the chemical signals are the neurotransmitters that get released. And so an action potential, like I said, travels within a neuron. That's represented here, action potential. And at the end of the neuron, at the end of its axon, it releases chemicals called neurotransmitters. And those neurotransmitters can cause an action potential in the next neuron, which would release neurotransmitters, and can cause an action potential in the next one, and so on. And so really it's an alternating electric signal within a neuron, but chemical signal between neurons. So last time we learned about just the electric signal, the action potential. Today we'll learn a little bit more about the details of action potentials and myelin, and then we'll move on to how this chemical signal works, the neurotransmitters and the synapse, or where the axon meets the dendrite from one neuron to another. Now, last time when covering action potentials, there was so much detail and so many diagrams that needed to be drawn that I ran out of time and board space and didn't get to the final piece of that puzzle, and that is saltatory conduction. Saltatory conduction refers to the fact that the action potential isn't continuous along the axon, but that it skips or it jumps along. The reason it does that is because the axon is insulated with a protein and lipid layer called myelin. That myelin is provided either by oligodendrocytes in the brain and spinal cord or Schwann cells in the peripheral nerves. And so here you can see the myelin covering, but it's not a continuous myelin covering, it has to have gaps. And those gaps are called nodes of Ranvier. And this myelin insulates against electric current, and so the electric current has no choice but to jump from node to node. What that does is it makes the signal travel more quickly, and it also lets the signal travel with more fidelity, or it's more reliable. So now we're going to take a look at this image, but blown up, to see the details. Now, this diagram should look familiar to you. It's very similar to the ones used to describe action potentials. There are sodium ions on the outside of the cell and potassium ions on the inside. There are voltage-gated sodium channels and voltage-gated potassium channels. The difference is that there's this large block here of myelin, and so I've shown dotted lines to show that the axon is inside and the myelin kind of wraps around it. So this part is the myelinated section, with this small section being a node of Ranvier, and this small section being another node of Ranvier. And so recall that what happens in an action potential is that these voltage-gated sodium channels will open and let sodium in. And we said before that the increase in positive charges kind of goes downstream and causes other voltage-gated sodium channels to turn on or to open. And we always drew those here, but really, if this large section is insulated, the next voltage-gated channels are here. And you might think that's a long way for these sodium ions to travel, and you would be correct. However, recall that because sodium ions are positive and positives repel each other, when sodium ions enter here, they actually push all of the rest of the positive ions over. And so these sodium ions are going to cause potassium ions to all shift over, which is going to increase the amount of positive charge here, which is going to activate these sodium ion channels. And so here, all of the positive potassium ions have been pushed because of the positive sodium ions coming in, which would cause these sodium ion channels to open, which would then let sodium in. And these sodium ions would then push potassium ions through the next myelinated section, and so on. And so the electric signal is really moving very quickly because instead of activating ion channels just next to them, they can activate ion channels that are way down here. And it's in that way that the myelinated portions can help the message move much more quickly. Now, there's an autoimmune disease called multiple sclerosis, and what happens there is that the immune system attacks the myelin covering and degrades it. And so it would be if you lost this myelin covering, underneath the myelin there's not a lot of the voltage-gated ion channels, and so the signal would kind of fizzle out here, or if it moves at all, it would move much more slowly. And that is why the symptoms of multiple sclerosis can be so varied. It's because sometimes there can be numbness because the neurons affected might be bringing in information, they might be sensory, or the nerves might be ones that are sending out signals to cause an action, and so there might be some paralysis. But it's all because of the myelin being targeted by the immune system. Now, hopefully you've heard the word synapse before, or synapse firing. And what that really means is it's the gap between one neuron and another, the axon of one and the dendrite of another. Those are called pre and post synaptic neurons. And when they talk about a synapse firing, the synapse is releasing neurotransmitters to send a signal from one neuron to the next. And so here we have a diagram of one at rest. This is the presynaptic axon, or the one sending the signal. And this will be the one receiving the signal, the postsynaptic dendrite. These are both parts of neurons. We're going to introduce some new colors for color coding. Calcium and related things, so calcium ions would be the plus sign. And these voltage-gated calcium ion channels, they are going to open up in response to voltage from an action potential, and they would let calcium ions through. These are chemically gated ion channels, and what they would do is that when a neurotransmitter lands on them, they would open up. And so they are chemically gated instead of being opened and shut by voltage. They are opened and shut by the presence of chemicals, neurotransmitters. Here, this is called the axon bulb. And these bubbles are vesicles containing the neurotransmitters for storage. And we'll see in a moment how they are triggered to release those neurotransmitters into the gap, the synaptic cleft. The process of the synapse firing involves a few steps. And depending on the resource you use, it might be more or less. Sometimes some of the steps are more expanded on and broken into smaller steps. Sometimes some are blended together. I've chosen to split the difference and make this into 5 steps. Step 1 would be the action potential reaching the end of the axon bulb. And an action potential, remember, is really just an influx or an in-traveling or an inward movement of positive ions. And to continue with the color coding from last video, I'm showing that with the green sodium ions entering the axon. That positive inward motion of the sodium ions causes the voltage to change at that area, and that voltage change causes voltage-gated calcium channels to open. Remember, voltage-gated means that they are controlled, opened and closed by certain voltages. And so the action potential reaching those voltage-gated calcium ion channels causes them to open, which would let calcium enter the axon bulb. When in the axon bulb, what calcium ions cause is they actually trigger the vesicles or the bubbles containing the neurotransmitters for storage. It causes those vesicles to fuse with the outside membrane of the axon. And what that does is it releases the neurotransmitters into the synaptic cleft. And now, once in the synaptic cleft, the neurotransmitters can migrate or diffuse over to the chemically gated ion channels that would receive them. And the neurotransmitters, when they bind to those chemically gated ion channels, will cause them to open. Now, on purpose, I didn't say what kind of ions they would be, because depending on the neurotransmitter and depending on the dendrite, there might be different ion channels, and they might even let in some negative ions. So far, we've talked about ion channels letting in positive ions, which this could be true in this case as well, but they also might let in negative ions. Now remember that the whole reason of sending these signals is to maybe cause an action potential in the postsynaptic cell. If the neurotransmitter causes an ion channel to open that would let in positive ions, that might start to excite that next cell. And if enough positive ions enter, then it could cause an action potential. However, there are inhibitory neurotransmitters as well, and those would cause negative ions to enter the cell, which would bring the voltage more negative and cause it to take more of a signal to cause that next cell to have an action potential. It would inhibit an action potential in that next cell. So excitatory would let in positive ions and cause an action potential to be more likely in the next cell, whereas inhibitory neurotransmitters would cause ion channels to open, which would let in negative ions, which would make it more difficult or less likely for an action potential to happen in the next cell. Now, lastly, this signal needs to stop. And sometimes the chemically gated ion channels actually have a function of breaking down the neurotransmitter chemicals, and then they would close themselves. Sometimes the neurotransmitter chemicals just fall off and are reabsorbed into the axon. Once the signal needs to stop, the neurotransmitters are either broken down or reabsorbed, then everything would kind of reset based on similar functions that we saw last time, where the sodium-potassium pump would be working, the neurotransmitters would either be created or reabsorbed and reset into vesicles, the calcium ions would be pumped out, etc. Well, again, thanks for being here. I hope you enjoyed learning about the saltatory conduction and how synapses work. I hope that you're able to understand it and incorporate it into your own work. Next time, I'll be using similar images to explain how psychoactive drugs work on the nervous system and the different ways that they target synapses. So I hope you enjoyed it. I hope you understood this so that you'll be able to understand that later on.